The core for Applied Genomics and Ecology at the University of Nebraska-Lincoln provides 454-based next-generation sequencing services to its clients and uniquely offers a microbiome analysis database designed to accommodate the client's 16S data in a user-friendly format. In this video, I will demonstrate how to use this database. So, to begin, navigate to the CAGE homepage at http://cage.unl.edu. You can also Google us. From here, click on CAGE Microbiome Analysis Database. Users who are regular CAGE clients have unique login and password combinations generated for them. And for this guide, though, I will log in as a guest, which will give me access to a public set of data that we have released as a demonstration set. And if you want to follow with me, you can also log in with this combination. Login is guest and password is anonymous. So once you log in as a client, you will see an experiment code and a blank screen right now because nothing is selected. Some clients have more than one experiment. That is why we have a selection menu here. So you begin by choosing your experiment code and clicking on the yellow filter button, which will then retrieve the records from our servers. At the bottom, you will see the total number of records and they're all in their status, cleaned and filtered. The records, in addition to giving you the experiment code, obviously give you samples, which you can select some samples that you want, not the others. Type of sample can be more than one. Regions of the plate on which they have been sequenced. There's always 2 of them, so you may select region 1, for example, which will narrow it approximately by half. Primer set, there also may be more than one. In this experiment we only used one primer set. Barcode ID, and then if you scroll over to the right, you will also see dates of sequencing. Now I do have 320 samples here. Obviously they were not all sequenced on the same date, so if I know more or less what I'm looking for, I can select the dates from here or scroll over some more, and here are the R directory names. These are the run directory names generated by the machine, which also have the dates incorporated, year first, then month, then date. Let's say I want this one. Okay, so now I have, remember I selected region 1, so I am narrowed to, only limited to samples from region 1. That was 43, and they were all from one run. This was the run on August 19th, 2009, for example. Now here I can also see the number of reads for each sample, the longest read length in each sample, and the shortest read length in each sample. We also provide FASTA and quality files for each sample. These can be downloaded by clicking on them individually if you want individual samples, and then it opens as a text file, and you can save as or save page as if you're working in Explorer. Select your directory and save it as a text file. And these will give you the quality scores. Or if you want the actual sequence, here you go. Same format, save as, choose your directory, save, and close out. Obviously, if you don't want to be clicking on them by hand and you want to download all of these together, what you do is hit this little check mark. This will select all samples that are on this screen. If you want to narrow it down some more, you may choose certain barcodes, or there's really, in this example, no other ways to narrow it down, but in other instances there are. Then in this box, give it a file name, that you want it to be saved as. For example, all records, and then choose your format. Do you want the system to download only the FAST files, only the qual files, FAST and qual together, or in comma delimited format, all of the information? And in that case, it will open with Microsoft Excel. And I'll just open it. So the comma delimited file will basically download this screen in a sortable format. Let's see my metadata if you need it. Now, to send records to classifier algorithm, you can select all records and then hit the little blue icon, which will open a gateway to our pipeline to a classifier. Make sure you have the files that you want, the samples that you want. These numbers, this is the first number is the experiment name, then the sample name, then the region on the plate was 1, remember, and the primer set was also 1, and then the barcode ID. This is how these names get generated. Can enter your username however you want, and your email address to which you want this data to be sent. I'm going to enter mine here, for example, and then you click submit. When you receive the data from classifier, it will come to you in this format from fangurima@gmail.com, and the classifier proportion results. This is one of our experiments. And 6 output files are generated. One is proportion file, then proportion 1, proportion 2, mapped hierarchy, hierarchy assignment file, and sample FASTA files and above files archived into one zipped archive. We usually use these. The prop1 file calculates the proportion of every taxonomic unit in relation to the total number of reads in the sample. Now when you download this, and I'm going to transfer this from my email program into my internet window, this is basically a TXT output. So you would want to save page as, and you can save it as a text document and then import it back into Excel. And what you will see is that classifier is a bacterial database, so most of my